Today we will try to discuss some of the theories of carcinogenesis, how the cancer developed. Of course, in the earlier days, people had different opinions, like especially in the time of Hippocrates and Galen, it was thought to have humoral origin. They called it as black bile. Humor, basically, they meant body fluids. They thought that body had 4 humors, that is blood, phlegm, yellow bile and black bile. When these body fluids were kind of balanced, a person is thought to be healthy. Too much or too little of any of these fluids will cause a disease. So, this was the thinking. An excess of black bile in various body sites was thought to be a causative factor for cancer. So, this was the idea. Of course, this theory of cancer was passed on by the Romans and was accepted even by that Galen. Peculiarly, this remained kind of unchallenged for nearly 1000 years. Then came what is called a lymphatic theory or lymph theory. Stahl and Hoffman were the people who theorized that cancer was composed of fermenting and degenerating lymph varying in density, acidity and alkalinity. The lymph theory also gained some support. John Hunter, who is said to be responsible for this kind of a theory later also after Stahl and Hoffman, agreed that tumors grow from lymph constantly thrown out by the blood. So, this was the idea in those days. Of course, let us not worry about the historical theories. Let us start understanding the latest or modern theories. One such theory that is widely accepted is called somatic mutation theory. I kind of told you about this already. That is, a mutation will initiate changes in the normal cell and several mutations will occur, and this is in the body cells, not in the germ cells. And then this will trigger such an environment that finally the cells will be converted to cancer cells. This requires several mutations, not simply one mutation, at least a dozen or more mutations are required. So, this is the kind of somatic mutation theory. We will look into it. People also believed in what is called clonal hypothesis. For example, you take the normal cells, something like this, and let us say some change occurs through genetic or epigenetic modes, and that will result in change in the structure and function. Let us say that you see this something like a light blue and a dark blue cell is formed. That means some changes have happened, mutation or something. So, this produced one clone. Now, this will upon cell division will produce other clones. So, this is like monoclonal. One cell is altered and when it divides, it produces its own clones. So, this is one idea, but this is not the same. So, like that, in several portions, several clones would have formed. So, there will be such monoclonal tumor cells, 2 or more. So, when they combine in a tumor, in a given tumor, you get so called polyclonal tumor cells. All the tumor cells known now are actually heterogeneous. So, they are like that. They might have originated from a single cell, it is possible, but many such single cells which had some mutations would come together to form a polyclonal cell. So, this is the theory, somatic mutation theory. There is another theory called tissue organization field theory. Interestingly, you see, none of these theories will explain all the features that we find in cancer cells. That means we have not understood it completely yet. What happens is certain theories will fit certain situations. Now, all of us are born by the fusion of sperm and ovum, and then that forms a zygote, and from that you get the fetus or whatever, right. So, that we know. Now, all of you know what is a genome. Genome is the total genetic information present in a given cell. Now, when you have this differentiation, when you get liver cells, heart cells or bone cells or whatever, what is the difference in genome? All of them have the same genome, right, but a liver cell is a liver cell because it expresses, let us say in human genome there are about 20 to 30,000 genes. All these genes are present in all the cells, whether it is a liver cell, brain cell or bone cell. Then why they are different? That is because the gene expression is different, right. They express certain set of genes out of these 30,000. Liver cell expresses a certain set of genes and bone cells express a certain other set of genes at a given condition and cells, and so they function differently also. So, if that is the case, then carcinogenesis is a problem of tissue organization. That is, simply if you change the expression, you change the function and the morphology. That is, from one zygote you are getting a liver cell, you are getting a kidney cell, you are getting a bone cell. So, what is happening? It is not mutation. So, without mutation also a normal cell can be changed into a cancer cell, correct. So, this is the idea, that is the idea of the tissue organization field theory. They do not agree, people who advocate this theory, they do not advocate mutation as the causative factor for cancer development. They think that a simple change in expression of genes could result in change in the structure of the normal cells into cancer cells. So, that is one idea. So, they say that it is a problem of tissue organization comparable to organ formation during early development. That is what I just mentioned to you. So, this is one of the ideas. So, why should you need that? Stem cells also, they can give rise to other types of cells. So, is it like mutation is happening there in stem cells per se? So, why not? So, that is the one idea. But then practically if you see, all cancers have multiple mutations and genomic instability. So, many scientists think that fits with their reality. So, somatic mutation theory seems to be acceptable to many people. So, that is what they are looking at. In between, of late, there is what is called a cancer stem cell theory. You know what is a stem cell? So, the normal stem cell, it can give rise to the progenitor cells, and they may differentiate, and they may give rise to some kind of some genetic changes should happen. Here also you see genetic and epigenetic changes may happen, and that may produce the so called cancer stem cell, but in terms of number it is a minority. Only very few cells are cancer stem cells in a primary tumor. Once you give treatment to the primary tumor, many of the cancer cells are dying, but these cancer stem cells, which are a minority as I told you, are in a quiescent state. Quiescent state means non-dividing state. They are sleeping, dormant, something like that, and they are unaffected by these chemicals or radiation. So, they escape this treatment and they remain like that for a number of years, and then when the microenvironment becomes conducive, they may again give rise to tumor. So, this is what is the cancer stem cell theory talking about. So, you see, there are differentiated cells which under some stress conditions or they may get de-differentiated, and some genetic changes also, genetic as well as epigenetic changes may help in getting cells which are having stem cell like properties. So, this is also possible, and then they may also be converted to cancer stem cells. So, cancer stem cells may arise from normal stem cells or from other differentiated cells also. So, they can have different origins. These are thought to be cancer initiating cells. Many people believe that, but some people do not agree with stem cell theory also, because again as I told you, this does not fit with all types of cancers. Some cancers you can explain this, but not all types of cancers. So, that is why no theory helps you to understand. Again, another interesting question is, okay, you understood that the cancer cells are formed from normal cells by certain mechanisms, maybe somatic mutations, maybe through that tissue development kind of thing due to changes in the expression, epigenetic and genetic changes. But how is that they move from one tissue to another? That is a $1,000,000 question, which means we do not have an exact answer even today, although we have some theories as usual. One of the theories is what is called anatomic model. Secondary tumors occur in the organs which they come across first, that is neighbor. See, when you want to fight with the neighbor first, right? Then only the distant people you can, first you have to invade the surroundings, then only you go to other places. So, that is one idea. So, whatever tissues they come across during their journey into the blood, they may invade that tissue. So, tumor cells from colon, for example, as I told you, this again does not fit with the other cells, other tumors. If you remember, I told you the breast tumor, the secondary site is something like brain, bone marrow. So, where is the neighborhood there? Where is breast and where is brain, right? They are anatomically at distant places. So, this theory does not fit all the cancers. But what is fitting with this theory is the colon cancer. Colon cancer, first the cells pass through capillary bed of the liver, and so the secondary site could be liver. So, that fits in suitable environment for the growth of secondary tumors. If they are present in the liver, then they will settle down in the liver. So, this is what the anatomic model says. In somewhere in, if you trace the history, Stephen Paget, it is called Paget's hypothesis also, somewhere in 1889, he observed that breast cancer patients often had secondary metastatic tumors in the liver. For him, the selective accessibility of the liver by the blood supply was not acceptable. Anatomic model he was not accepting. It is not that you are a neighbor and so you these cells have spread to the neighboring tissue, but selective accessibility is what is important, was not acceptable to him, and as many organs are equally accessible, it is not only a liver, but many other organs may be nearby, why it is going to a selected tissue. So, he thought, just like if you think seeds, seed and soil theory sometimes they are calling this. Why? Because if you take seeds and take it in a drone, for example, and spread it in our campus, do you think all the seeds will grow? Suppose the seeds fall on a rock, do you think they will grow? So, they require suitable soil, water, light, all those conditions. So, like that, if cancer cells can spread through blood, to different organs, they will definitely not grow in every other organ they encounter. They will be selective, because a selective environment is needed. So, that this is called seed and soil theory, but this does not explain what is that environment, what is that selective environment that will allow them to grow. So, this was difficult to understand why metastasis occurs. So, as time passed, people have thought of many molecular theories. It is very interesting. Tumor cells in the blood and lymphatic vessels have equal access to organs, all organs, but multiply only in those organs that have appropriate growth factors. This is one theory. So, you must have growth factors. There is another theory that thinks about endothelial cells lining the blood vessels in the target organs express certain adhesion molecules. So, they put something like a brake. The cells are flowing in the blood and they will come across these adhesion molecules and they will stop there circulating tumor cells in those organs, and then they will penetrate that region and they will grow there. So, this is one theory. Again, as I told you, this is only a theory. What is more attractive is called chemo attraction theory. You can see here normal cells, and then they become malignant and they travel through the blood, and then they have certain chemokines, the attractant molecules enter the circulation, they attract the circulating tumor cells containing the receptors. What it means is, the cancer cells have certain receptors and certain tissues express the corresponding ligands, and so they will only go to those tissues which will have the chemokines or the ligands. So, you have the receptor on the cancer cells, you have the ligand on the target organs. So, it is now understandable that they will be attracted only to those organs, and that is why they go to lungs or bone or brain and so on. So, they are said to be the preferred secondary sites. This stimulates them to invade the walls of the blood vessels and enter the organs. One such example is what is called CXCR4 receptor in breast cancer tissue. It is highly expressed in breast cancer tissue compared to normal breast tissue. Its corresponding chemokine, the ligand, is called CXCL12, which is a chemokine. It is expressed in many metastatic sites or organs of breast cancer, such as lymph nodes, bone marrow and lungs. So, it is because of this reason breast cancer spreads to these organs. So, that is the idea. So, this is one acceptable theory. Now, I have already told about this. The conventional therapies like surgery, radiation, chemotherapy, they all have side effects. So, that is why we need an understanding on the molecular biology of cancer cells, and most of the modern research is focused on this kind of an understanding on the molecular aspects of cancer cells, different cancers, and they developed what are known as molecularly targeted therapies. Because if you know what are the targets in a particular type of cancer, you can target them. So, that is it. There is another therapy called combination therapy, because some of the molecularly targeted therapies may not be standalone therapies. Standalone means that therapy alone is sufficient to cure cancer. Many of the conventional therapies are like that. You do either do surgery or do radiation or do chemotherapy, but you can combine them also in certain cases. That is called combination therapy. For example, you may not be able to cure something by radiation may not be accessible to some sites. if you want to do that, you have to penetrate. So, you may combine a little bit of radiation you may give, and then you can do surgery, and some cells which are remaining you can kill them by radiation, for example. Like that you can combine chemotherapy with radiation, or you can combine chemotherapy with some of the molecularly targeted therapy. So, this kind of therapeutic options are gaining importance these days. So, we have discussed what is cancer, what are the different types of cancers, what is the cancer burden in India and the world. We have given an overall idea about the molecular basis of cancer, causes of cancer, and we also discussed what is metastasis, what are the theories of carcinogenesis, modern theories, and we just touched upon the conventional therapies.